So I'm one of probably 3 or 4 people in the world who've worked on CRISPR for more than 10 years. And I've seen CRISPR rise from almost nothing, a singularity in the genomes of bacteria and archaea, to one of the hottest topics arguably in science today, and for not just years, but likely decades to come. So CRISPR is a very compelling, very interesting, highly valuable technology. And what I'm here to talk about today is the CRISPR craze, because it is crazy in so many ways. Elizabeth Panizzi in Science coined that in 2013, because the CRISPR craze was starting then, and it has done nothing but grow ever since. So it's very, very compelling, and it's crazy in many ways. And what I'm trying to do today is walk you through, where CRISPR comes from, what CRISPR is, and how it's being used. So I'm going to take like 4 segments of 8 to 10 minutes and try to tell you everything you need to know about CRISPR for the next few years, likely for the next few decades. So I'll start with like basic biology of CRISPR. What is CRISPR from a scientific standpoint? What is it as a phenomenon, as the adaptive immune system in bacteria? And then I'll walk you through how the machinery from what's called CRISPR-Cas systems has been repurposed and repackaged as what people call CRISPR tech, or CRISPR technology, or a tool that is enabling what's called genome editing. And then what I'll do is I'll walk you through a number of applications focusing on microbes, but also extending beyond this, and walk you through where we are today in the terms of genome editing 2.0. So the golden age, arguably already, of genome editing and the opportunity that CRISPR techs afford for genome editing. And then last but not least, I'll start the conversation about the CRISPR craze, about how disruptive this technology is from a technical standpoint, from a business standpoint, from an ethical standpoint, from an intellectual property standpoint, from a business standpoint, and from a consumer standpoint. And we can wrap that up with a series of implications, not just for scientists, not just for educators, not just for academics, but for industry, for consumers, and for patients. And for people who care enough about CRISPR to be here today. So we'll see where that takes us. All right, so let's start with the science. I'm a scientist. I went to school a lot. I got a bunch of degrees from different schools all over the world. And the reason I did that is because science is cool. So in my mind, science is cool. It's not really work. It's a lot of fun. It's work sometimes when you have to teach, right, give exams and stuff like that. But the science itself, and the science of CRISPR in particular, is really fun and is really interesting, and I'm going to try to walk you through that for just a few minutes to give you enough knowledge to talk about CRISPR. So this is what CRISPR looks like in nature, right? Like gibberish. A, T, C's, and G's, right? And CRISPR is actually an acronym that stands for Clustered, Regularly Interspersed Short Palindromic Repeats. So that's what CRISPRs look like in nature. And actually, 99% of people who use CRISPRs have never seen a CRISPR. Anybody see anything? Cross your eyes a little bit. What do you see?
Diagonally.
Diagonal, right? Which way is it going? All right, that way. Okay? So that's what you should be seeing. I'm going to help you a little bit here. And if you're not NC State right-averse, which nobody should be here, right? This is what you should see. Clustered, because they're all together in the genome, regularly interspaced, because they're spaced at an exquisite periodicity in terms of size, short, because those DNA repeats are 30 to 36 nucleotides, depending on the organism you have and the CRISPR-Cas system that you have. P stands for palindromic. They're partially palindromic. For those up front, you can see the G, T, 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 the 5' end is the reverse complement of the sequence of the 3' end. And then R for repeats, because they are DNA repeats. That's all we need to know about CRISPR. They are weird-looking DNA repeats in the genomes of about 46% of all bacteria in the world and about 90% of archaea. So they are widespread, weird peculiarity from a genomic standpoint. And what they are in many ways is the bacterial equivalent to adaptive immunity in humans. So let me take a minute here. On a show of hand, who's been vaccinated before? Right? Like you're here because you're smart, right? And you've been vaccinated before. And essentially, the basic concept behind vaccination in humans is hinging on the interaction between antibodies and antigens. So this is maybe like middle school biology now, maybe high school biology now, right? You have a lock and key mechanism whereby your immune system will generate antibodies that recognize stereophysically antigenes, antigenic elements that are not self, that are foreign elements. And this lock and key allows humans and mammals and others to actually recognize physically non-self and distinguish from self and then attack it and mount an immune response. In many ways, what CRISPR is conceptually is the equivalent of an antibody-antigen molecular interaction for adaptive immunity, but rather than be protein-protein driven, it's nucleic acid driven. And rather than have a lock and key mechanism, the way CRISPR works is by having a DNA sequence like the one I just showed you that will tell the bacterial immune system what to recognize as non-self and then cut it. Cut that DNA in a sequence-specific manner, because each genetic element on planet Earth has peculiarities in terms of their sequence content, and then any given word, a combination of A, T, C's, and G's, can be programmed or can be sequenced or can be defined as a unique target, and CRISPR is a system that enables bacteria to cut DNA and recognize foreign DNA. That's all you need to know. And there's only 3 things you need to know about CRISPR. Number 1, it's DNA encoded, so it's a DNA sequence. Number 2, it's RNA mediated, so the central dogma of biology says DNA goes to RNA, goes to proteins. So CRISPR DNA is transcribed into CRISPR RNA, and this CRISPR RNA, cRNA, will tell a pair of molecular scissors called Cas, CRISPR-associated sequences, to target that word and cut it, to recognize a unique DNA type word in the genome of a virus or an invader, and then cut it selectively and precisely. That's all you need to know. DNA encoded, RNA mediated, DNA targeting. Now, in reality, there's a lot of different types of CRISPR-Cas systems in nature. So think of all the tools you have in your kitchen. There's different kinds of knives and slicing contraptions to do different kinds of jobs. So in many ways, most of the popular CRISPRs are like a knife right here, where you can take a CRISPR and you can slice a piece of DNA, like a fillet. There are some CRISPR-Cas systems that are more like a chainsaw or a blender. They're going to chew up DNA. Others are shredders. They're CRISPRs that will shred DNA or RNA. And others cut it in an offset pattern or just do both the cutting and then shredding. So you have different kinds of CRISPR-Cas systems that are all DNA encoded, RNA mediated, DNA targeting, but they recognize and cut and process and cleave DNA in different ways. And the way they work is essentially the same. It's all DNA encoded, all RNA mediated, all DNA targeting, but how they go about doing the cutting and what kind of cutting outcome they get varies depending on the Cas machinery that goes with it. The CRISPR-associated sequences, the Cas proteins that do the job. So the CRISPR is what tells the Cas what to cut, where to cut, when to cut it, and how to cut it. And the Cas is the molecular scalpel that does the cleavage itself. All right, so let me show you a little bit how it works in an animated manner. So imagine you just had pizza for lunch, you're a bacterium, you're laying down, taking a nap, and you're a single cell. So this is all of you right here. And you're lucky and very well endowed with a fantastic CRISPR-Cas system. There's Cas genes, the CAS on the left. Each arrow is one gene, and each gene encodes one protein. And there's the very famous or infamous Cas9 here on your left, which is a very popular and potent molecular scalpel that can cut DNA precisely, accurately. Okay, so Cas9 is a DNA slicer. And then to the right of those arrows and those genes, you have a CRISPR array. Again, clustered regularly interspatial palindromic repeats, and each repeat is shown as an unlabeled black diamond, because all the repeats are the same. Repeat, 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 repeat. And in between any two repeats, you have a unique piece of DNA from a unique vaccination event called a spacer. So I can see from here there's 33 repeats and 32 spacers, and the spacers are numbered right to left, not to confuse you. They're numbered right to left because they're like a CRISPR train. The front of the train is the first car that's leading the way, and then you add a second, and a third, and a fourth, and a fifth. And every time the bacterium is exposed to a new virus, it's going to copy and paste a piece of that DNA as a new car in the CRISPR train. All right? So you've been vaccinated 32 times, and there you are enjoying your pizza, laying down, taking a nap, and you make either a bad decision or you're out of luck, and some random virus is going to come and attack you. That virus is going to insert and inject its DNA into the cell, and the purpose of the viral DNA is to take over the host and replicate and multiply. Okay? Now, lucky for you, you have CRISPR. So the Cas machinery is going to recognize this as non-self, an invasive nucleic acid, an invasive non-self DNA. And it's going to scan that DNA for a short sequence motif that it has very high biochemical affinity for. So something that can latch on to. So imagine CRISPR-Cas being like one of your hands. It's going to look for something it can latch on. Some kind of appendage, some kind of sequence it has a high affinity for. And then it's going to copy and paste a short piece of DNA that's right next to it. So think of it as, if you guys have phones, like a Snapchat. It's going to capture, right, a short piece of information from the DNA it just latched onto. So you grab someone, pull out your phone, and take a picture. Or if you're a bit older, like a Polaroid. Right, if you're very old, like, you're going to draw it out, right, like a wanted sign. And it's going to attach this piece of DNA, this new spacer, to a repeat, a CRISPR repeat, creating a repeat spacer unit. And it's going to insert this genetic information on the left side, called the 5' end of the first repeat here. And it's going to insert, essentially, vaccination event number 33. It's going to insert a novel piece of DNA from the virus that infected it, and essentially constituting a time series of vaccination events. This is like a genetic vaccination card. So this tells the system, and it writes in the DNA of the bacterium, every piece of DNA of every invasive element that it has seen over time, things like plasmids in bacteria or viruses in bacteria called bacteriophage. I should say bacteriophage, this used to be French. And he's going to insert that into this CRISPR array and add on to his collection of vaccination events. So it's like me teaching at NC State and having students who go on spring break being able to tell, well, last week you made a bad decision, there's a new viral sequence in your genome. And I can compare and contrast this vaccination sequence to your neighbor or your roommate or your cousin or your sibling or your parents. And I could be able to tell whom got vaccinated against what, in what order, and at what given point in time. So it's a vaccination record in DNA. Once you're vaccinated, DNA encoded, then this thing gets transcribed as CRISPR RNAs and processed into small pieces of RNA that each contain the genetic information from one vaccination event. So it's a short word of DNA that tells the system, this is the sequence that I've been exposed to before from a virus that infected me. And then it's going to recruit the Cas9 molecular scalpel to find something that matches this word, this DNA sequence, and then cut it in a sequence-specific manner. But the cutting itself is very precise. So he has this word, right, this DNA word that it's looking for in red at the bottom. Okay, and it's going to latch on using that hand to that sequence that it has an affinity for, and then it's going to count 3 letters, -1, -2, -3, and slice DNA precisely. So it's going to take that molecular scalpel, look for the word in the DNA of the virus, and slice DNA precisely 3 letters away from the end of the sequence that it captured. And then in doing so, you cut the viral DNA, and the virus dies, and the host makes it. That's all you need to know about CRISPR. DNA-encoded, RNA-mediated DNA targeting, and it's a sequence-specific viral cleavage mechanism. Now, the thing I just showed you took about 30 years to show by all kinds of people all across the world. Right? CRISPR was actually first observed back in 1987. Born in 87, that's 30 years old. And it wasn't baptized until 2002. It was nameless for 15 years. Okay? And it wasn't until 07 that we showed, actually in my group at DuPont, that CRISPR works as an antiviral system by picking up pieces of viral DNA. And since then, in the past 10 years, people have shown how it works, why it works, and people have established that this is a DNA sequence-specific cleaving mechanism that enables bacteria to escape viruses. So from a scientific standpoint, that's cool, right? But really, only people who care about bacteria care. So the reason CRISPR is so popular is not because of that. The reason CRISPR is so cool is not because it's an adaptive immune system in bacteria that enables things that make yogurt or cheese to survive phage attack. The reason why it's so popular and so cool is because this molecular scalpel can be used to do what's called genome editing. Okay? So what's genome editing? Exactly 4 years ago, my friend Jennifer Doudna, shown on the left here, she's the charming lady right here, showed that you can take that system, right, with a piece of RNA and 1 protein, and you can rewrite the word designed in the RNA to cut any piece of DNA you want. You can take the molecular scalpel out of a bacterium into a test tube like this, okay, and you can engineer and redesign and reprogram the guide RNA that tells it where to cut. That was step 1. So 1 pair of scissors, 1 molecular scalpel, and 1 word you can engineer and design and program to tell the enzyme where to cut. The second tipping point was from Feng Zhang, another good friend of mine, who showed that you can use that to edit DNA in humans. So how does that work? So who's used a razor before? Any razor? Come on. Right? Regardless of your gender, your genotype, your phenotype, at some point you've used a razor blade to cut some part of your body. I don't need to know. Trust me, I don't want to know. And occasionally that scalpel that you have in your hand is going to generate a cut in your skin. Right? You're going to cut your skin. And what happens when you cut your skin? You bleed. And what happens after that? It heals over. It's called repair. So when you cut your skin repair mechanisms are going to fall into place. And if you're a guy or a hockey player or a football player or a manly guy in the yard and you cut your skin, you're going to patch it back together. Put a little duct tape on it, put a little dirt on it, put a little tape on it, sew it back together, right? And you're going to have a little scar where you cut your face or your fingers or your leg or whatever, right? Or if you're heavy-handed or you're a little bit vain or both, you're going to go to the surgeon and say, I have a cut right here. I need you to repair it. And I want my left cheek to look like my right cheek. I want my left leg to look like my right leg. So you're going to give the surgeon a template to repair your skin and do a very small scar. This concept applies to genetics. So when you take a CRISPR scalpel and you cut DNA like this, right, you slice DNA, you generate what's called a double-stranded DNA break. You're going to slice DNA that way, right? The DNA repair pathways come in and repair it. And there's two main DNA repair pathways. One is shown at the top right, called NHEJ, non-homologous end joining. I'm going to take my two pieces of DNA that are non-homologous, and I'm going to patch them back together like duct tape, right? Or sew them back together. And when I do that, I'm going to have a little scar. That scar is a small insertion or a small deletion or a change in the letters of the DNA exactly at the site of cleavage. Alternatively, you can go to the genetic surgeon and use what's called HDR for homology-directed repair, and you're going to give the DNA repair system a template to patch that back together and rewrite that sequence exactly the way you tell it to, which means that the scientist, the investigator, right, can come in and then design a single letter change or insert a word or delete a word exactly at the site of cleavage. And that's what is called genome editing, because genomes are texts of DNA. And what editors do is they correct typos. They change a letter. They add a letter. They remove a letter. They add a word. They remove a word. You can add a sentence, remove a sentence, or a whole paragraph, or a whole chapter in the DNA book of life. It's that simple, but also that consequential. With CRISPR-Cas technology, you can take a very fine molecular scalpel, target any piece of DNA you want, cut it precisely, and rewrite the genome exactly at the site of cleavage. You can edit any sequence you want, just like that. That's the power of CRISPR. Now, this is the power of CRISPR circa last year. This is so 2016. Okay? What you can actually do is you can take this molecular scalpel and kind of dull the blade. And what you do when you dull the blade is you make it bind to DNA, but not cut DNA. And what you do when you do that, because it has such this strong affinity for DNA, is that it can bind to a gene of interest and then prevent other things from happening there. So if you're a biochemist and you've heard about transcription, you can block the activity of an RNA polymerase. It means that you can turn off the ability of a gene to be expressed or transcribed. Or you can tether Cas9 to an activator and you can turn on transcription. It's like turning the lights on or turning the lights off. I can't quite do that here. There's no button, right? In the expression transcription profile of any given cell of interest. Or you can tether Cas9 to an effector domain of interest and change the structure of the DNA, the stereophysical availability of a particular stretch of DNA. Or you can tether a fluorophore to it and unlighten the sequence of DNA. You can do that with 6 different fluorophores and do what's called CRISPR rainbow. You can visualize the location of DNA in any cell you want. Or Charlie Gersbach down the street at Duke showed last year, you can actually tether Cas9 to methyltransferases and acetyltransferases and alter what's called the epigenetic state of a sequence of interest. You can change the methylation pattern of DNA. So essentially right now, we can do what's called genome editing 2.0. You can change the sequence, you can turn transcription up, or you can turn it down. You can enlighten DNA, you can deliver effector domains, or you can rewrite the epigenome. The genome, the transcriptome, and the epigenome, you can rewrite in any organism you want, any sequence you want, any way you want. That's that. It's as simple as it gets. It's also, again, as consequential as it can get. And what's great about CRISPR is that it is, granted, a 4-year-old technology, but the most charming, the most powerful, the most endearing 4-year-old you've seen in your life. It's just unbelievable tech. Because it has all those advantages: programmable, specific, transferable, efficient, precise, affordable, quick, multiplexable, and scalable. You can order a kit to do CRISPR out of Addgene for $69, shipped in a couple of days. You can get one for Christmas, Valentine's Day, your birthday, or any given occasion. Right? You can tell your friend maybe you should change their sequence in their flawed genetic content. If they're scientists, they'll get the joke. Okay? So it's very powerful, but like any 4-year-old tech, it's imperfect. So this molecular scaffold I keep talking about is a little chubby, it's a little fat, it's a little hard to deliver to cells. So we've got to make it smaller, we've got to make it less biased, we've got to make it find all kinds of sequences that we want, we've got to make it specific, and more importantly, the true challenge is we have to make it more efficient. It's great to cut, but you've got to make it cut all the time, every single time, to edit 100% of what you want to edit in the organism you want to edit. So efficiency is the true challenge shortcoming of CRISPR right now. Delivery is important. So there's ways to improve that, but like any 4-year-old tech, it's going to get better. It is getting better every day, every week, every month. There's tens of thousands of labs working on this. Now, one of the things that we're working on is trying to do what's called navigating the genome. Can we very precisely go to any sequence we want in any genome and very selectively and accurately cut DNA right there? Right now, we're not quite there yet, but we're almost there. So we're working on this to navigate the genome and then optimize the stability, optimize the delivery, optimize the efficiency, and diversify the CRISPR-Cas toolbox. So you have all kinds of tools available to do all those things in parallel at the same time. So you need one tool to do upregulation, one tool to do downregulation, one tool to do editing, one tool to do epigenetics. So you can do it all at the same time in the same system, efficiently, selectively, accurately, precisely, and affordably. We're working on this. The toolbox is almost there. Now, the reason why it's taken the world by storm is because of what we call the CRISPR zoo. People in the last 4 years have been able to port CRISPR technology to all the animals I show you up here. All the model systems at the bottom, bacteria, viruses, model organism that scientists do to study and do scientific things. It's also been used in organisms used in food and ag. Right? You can see we're making CRISPR chickens. Not just crisp chicken, CRISPR chickens. Okay? It works for bacon too. Crisp bacon or CRISPR bacon. Okay? People have done the white button mushroom, corn, soy, cow. There's no limitation to what we can do from a scientific standpoint. And more importantly, beyond food and ag and science and research, the medical field have been overwhelmed by enthusiasm for scientists willing to use CRISPR to understand genetic disease, to cure genetic disease, to remove viruses, to do gene therapies, to grow new organs, to generate new species that can do anything we want them to do, almost just like that. Not quite the hand, more like a pipette click. Okay? That's taking the world by storm. And to do you an example, give you an example, to do it justice, right? People have shown you can take a butterfly, and butterflies, much like the human retina, have unique wing patterns. No two butterflies in the world have the same wing pattern. So people have shown you can take this butterfly right here, bring a CRISPR to it, target the yellow gene, cut out the yellow gene, and turn an offspring of that into a black butterfly with the exact same wing pattern. If you can do that, I'll let you think about what can be done. Arguably, the only limitation to what CRISPR-Cas technologies can do scientifically is the imagination of the user and the scientist, for good or maybe not. All right, so now that you know what CRISPR is and how CRISPR tech works, let's talk about how it's being used. So, anything that deals with genetics has been susceptible and has been CRISPRized, right? So research use, tools, kits, guides, enzymes, primers, plasmids, cell lines, softwares. Any researcher has access to a suite of tools and toys to play with and harness CRISPR technology, just like that. Get stuff delivered overnight. It's been used in bacteria, industrial biology and biotechnology, yeast and algae for food, biomanufacturing, household care enzymes, bioenergy, anything you can think of that's biologically relevant is being investigated and CRISPR enhanced. Obviously, plants and animals and microbes in ag, and in RTP, we have big ag, big time. We can talk about that. And then last but certainly not least is human medicine, gene therapies, antivirals, cell therapies, immunotherapies, xenotransplantations, microbiomes, antimicrobials, and gene drives against mosquitoes and invasive species for malaria, for example. So we can talk about that if you wish. Now let me talk about my lab for 5 minutes. Just my lab at NC State. One lab, 15 people. Right? And again, I've been using CRISPR for 10 years, but we are working on all kinds of applications of CRISPR down the street at NC State, a state institution, a land-grant university in the food science department to understand how those technologies can be used to enhance the food supply chain. So let me give you some examples, right? So we try to use this vaccination card to understand where bacteria come from and how they can compare and contrast to one another. Like I could go into this room and find out your vaccination card, and that would be a unique identifier of who you are and where you've been and what good or bad decisions you've made at what times in your life. It's very scary. Even if you had an identical twin, that could tell you guys apart. So we use those technology to track all the bacteria used in the food supply chain. So if you go to Chipotle tomorrow and you get sick, I can sample that and tell the CDC where the problem comes from. If you're a soldier and you're a victim of a biothreat, we can sequence that and tell the Department of Defense where that bug comes from. If you go on a cruise and you get sick, we can tell whether it's the same bug as last week or what port it got picked up at. So it's extremely powerful. So let me give you some examples. This is C. diff, Clostridium difficile. We can track C. diff all over the world and tell, just using CRISPR vaccination profiles, where bacteria come from, where they've been, and how they compare and contrast to other cousins we've seen before. So we can tell when somebody contracts C. diff where it comes from and how it compares and contrasts to other patients at the same hospital or the same town or the same county or the same country or the same continent. And we can backtrack and map this in time and space to rebuild the history of human disease. We've done this.
So when you're doing this, is this based on the unique binding of a specific Cas9 sequence?
No, actually, it's based on the unique series of events that have enabled the CRISPR, it doesn't even have to be Cas9, it can be any type we want, to have acquired unique signatures sequentially from invasive elements.
So can you discover something that you haven't seen before?
Absolutely, yes. Yes, it's agnostic. We can find strains we've never seen before. Actually, it's good if we've never seen before, so now we can tack it on to the database. Agnostic, yes. And we've used this to look at things like food spoilage. Right? So we can go down to Mount Olive and we can tell whether, Mount Olive pickles will be subjected to more or less spoilage because they have certain kinds of bacteria that are there. We can go to different farms in Wisconsin that have salmonella problems and tell whether neighbors have the same salmonella problem or not. And we can backtrack that to the food and or the feed and or the water and or their neighbor and or wherever they want. We can do that in E. coli, with CDC and FDA. We can do that with bacterial cultures used to make cheese. I'll get back to that in a minute. So it's extremely powerful, extremely potent. Another way to harness CRISPR technology for the food supply chain is to make the great cheese you guys all enjoy for the pizza. All that cheese you use today was made at NC State in my lab. No, I'm kidding. I'm kidding. Just relax. Okay? Keep it in. Have another bite. All right. So I've been working in the dairy industry for a long time. And in the dairy industry, bacteria that make the best yogurt and the best cheese oftentimes get hammered by what's called bacteriophage or bacteriophage, right? Because there's viruses or bacteria all over in the milk supply chain. Now, scientifically and conceptually, we could take the best strain in the world, right? So shown at the top right here, my friend with 32 spacers, and then select in the lab natural phenomena, natural vaccination events that enable this great starter culture that makes the best yogurt in the world to become resistant to this phage from Canada. And then do that again with the phage from Mexico. And do that again with the phage from Argentina. And do that again with the phage from Italy. And do that again with the phage from China. And we can do that in 3 different labs in the world. One in the US, one in Asia, one in Europe. We can blend those 3 cousins and sell those starter cultures anywhere in the world to make cheese and yogurt forever. All right, let me show you how that works. So imagine, until you get a new phage, we can talk about that. So imagine at the top is your best starter culture of all time. DuPont Global Culture Collection strain number 7,710. I spent like 10 years of my life working on this thing. And this thing gets hammered on the right, sensitive to a lot of phages. So we can expose this strain to a phage from wherever, select a variant that's vaccinated. Take that variant, expose it to a phage from a second location, and select a natural variant vaccinated against it. And do that again and again and again. And over the course of 2 weeks, almost anybody from my lab can generate a super vaccinated strain that will make the best Greek yogurt you can think of forever. I got promoted at DuPont 10 times in 9 years because of that stuff. Now, DuPont is a global starter cultures provider to all the dairy industry worldwide. CRISPR vaccinated bacteria have been used globally in the food supply chain since 2011. Let me explain to that what that means. If you've had one bite of pizza, like today, one cheeseburger, one bite of yogurt, or one nacho, anywhere in the world since 2011, you've consumed a dairy product that was fermented using a CRISPR-enhanced starter culture. That's why you're so smart and so happy and so healthy. Globally since 2011. I'll let you think about that. Now, at the same time, companies like DuPont or any company that's using bacteria for any bioindustrial processing now has the ability to use this system to expose a strain to a series of different viruses, yellow, blue, pink, red, and green at the top. And that particular vaccination order, yellow, blue, pink, red, and green, is unique and cannot be replicated randomly in nature in that order unless you've taken that strain from me. So if I find a product with that strain that you didn't buy from me, you're going to jail and or writing me a big check.
Can the bacteriophages strike back like the Empire antibiotic resistance in
So they try. So we've studied that for a long, long time, right? CRISPR is so efficient that when it works this way, the viruses disappear. The only chance is for mixed viral genotypes to recombine strategically the part of their genomes that get hammered selectively by CRISPR and then enhance mosaicism. That just delays the arms race. Unless you have new viral families coming in and migrating in from without all the time, viral genomes evolve to extinction. We've shown that, we published that. Now, there are anti-CRISPR systems, there are CRISPR escape systems, but there are means to harness and select the spacers that target regions of the genome that cannot be mutated because they are lethal and or dysfunctional. So this is the way we would do that. So very powerful. But nature does have ways to try to escape. Just got to show them who's the boss. All right. Now, one really cool thing to address what you just mentioned with antibiotic resistance is we actually 3 years ago at NC State found a way to turn CRISPR into an antibiotic. And we found a way to tell the CRISPR in the bacterium not to target the phage, but to target self. And to do that, we used a CRISPR that's like the chainsaw, the Pac-Man, the axe, not the one that has a little cut in the cheek that you can repair, the ones that will ablate your arm and your leg and your head. And we've shown that you can very proficiently, very specifically, very accurately, and very programmably kill bacteria. So I can take two bacteria that are 99.9% identical and choose that small percentage difference to just kill one, or the other, or both. We can use that to kill E. coli, working on this to kill C. diff, working on this to kill Salmonella, working on this to kill Pseudomonas. So now we can go into a microbiome and cut out specifically not the whole microbiome, not wipe it out and give rise to antimicrobial resistance, but selectively target the agent responsible for infectious disease. That's the future of medicine, enabling us to address antibiotic resistance. Okay? All right. So just at NC State, everything I mentioned today is being done. Right? Crop genetics, animal genetics, starter culture genetics, biotechnology genetics, and probiotic genetics. And we work sometimes in GMO, sometimes in non-GMO ways with all the biggest players in the world to take that to the food supply chain. People like DuPont, people like GE, people like the USDA, people like the CDC, people like the NC Biotech Center, people like Pioneer, and others, to improve the genetic content of all the organisms of interest to the human food supply chain any way we want. And part of the challenge is to pick the right tool for the right job, to pick the right knife, the right scalpel, the right axe, the right Pac-Man, the right chainsaw to achieve what you want to achieve. Do you want to kill pathogens and eradicate them and wipe them out? Or do you want to selectively improve one piece of gene to enable cheese cultures to withstand phage attack? Different strategies, different problems, different solutions. Use the right tool for the right job. Okay? All right. Now let's spend 5 minutes talking about the CRISPR craze. This stuff is crazy. It's crazy. I gave my first talk on CRISPR 10 years ago. At the time, the whole CRISPR literature was 27 papers. I knew it all. And within one PhD cycle, depending on what school you go to and how good you are, right, we went to one paper a month, to one paper a week, to one paper a day. Last year, over 2,000 manuscripts were published on CRISPR technology. This year, at current pace, we're going to exceed 4,000 from just 27 total 10 years ago. Exponential rise of CRISPR. CRISPR is making the cover of a major scientific journal magazine every week. And this is just the start. CRISPR is getting prizes and awards and recognition and PR. It's crazy. Scientifically, the most disruptive technology since PCR, quantitatively. Okay, so this is the rise of the CRISPR papers. This is the rise of CRISPR shipping. So Addgene will sell you and send you a CRISPR plasmid, not-for-profit organizations only, for $69. Last year, actually 2 years ago, Addgene sold 24,000 plasmids. That's 2,000 a month. It's 500 a week. It's 100 every day. 100 new labs every day get their hands on CRISPR. 2 years ago pace. This stuff has been shipped to 81 countries. When you go home tonight, take a sheet of paper and write down the names of 81 countries that have CRISPR. It's mind-boggling. In breadth and depth and globalization, what I call the democratization of CRISPR from a scientific standpoint. Now, however cool it is scientifically, some of the biggest CRISPR craze is the news, is what I call the media craze. CRISPR made the cover of Time magazine on their July 4th issue last year, the most read and the highest volume issue for the year. That's not random. CRISPR made the cover of my beloved The Economist. Science never makes the cover of The Economist. Okay? So, there's over 172,000 news articles on CRISPR, 186,000 videos on CRISPR, and 6 million hits online for CRISPR. CRISPR is so crazy, somebody put my car on Twitter. Like I can't park at RDU quietly. It's crazy. There's CRISPR movies being made. CRISPR documentaries being made. CRISPR is on TV in the X-Files, right? And Luke Cage. Every week, there's an article, Wall Street Journal, New York Times, NPR, Forbes, on CRISPR. Very legitimate news feeds, FYI. All right. Now, however cool the media craze and the scientific craze are, as a MBA and business school graduate, an industry guy for 10 years who was global R&D director at DuPont in a division, I can tell you that this is where the biggest scale craze has occurred and is occurring. At the same time, you have multiple industries craving the same tech. Whether you're pharma, or biotech, or food, or ag, you're in. Whether you're a Fortune 10 company like a GE, a Fortune 50 company, a mid-cap company, a small-cap company, a startup company, or even a not-for-profit company like Addgene, you're craving the same tech. It's very rare for one technology at the same time concurrently to compel multiple segments and multiple types and sizes of businesses. This is the power of CRISPR. And to illustrate that, just in the past 12 months, 3 different startup companies based on CRISPR tech have gone IPO. One of which I'm affiliated with, Intellia Therapeutics. Disclaimer. That company, in less than 2 years, went from an idea to a publicly traded company at the NASDAQ. And today, I checked this morning, the valuation of those 3 companies is in excess of $2 billion. $2 billion. 1 year. Think about that. Now, this comes with caveats. Maybe we can talk about the CRISPR IP, what I call the French bakery paradox, if you're curious. People want to eat it all. It's crazy. They want to have it all and stir their face with everything CRISPR. The good, the bad, and the ugly. But as a state employee and professor at a land-grant university, with those great opportunities come great responsibilities. As a scientist, we have and we're compelled to be mindful of the ethical implications, the PR implications, the commercial implications, the regulatory and political implications. I'm on the faculty on 7 different departments at NC State, including the Genome Engineering and Society Center. We have to be reasonable. We have to be ethical. We have to define whether or not and how we can edit the human germline. How democratized this technology should be or not. We've got to engage in a dialogue with all the key stakeholders, including the public, to define the guidelines for scientists to operate and use that technology. By the way, not just here in RTP, not just here in North Carolina, not just domestically in the US, but globally. It's our responsibility. And by the way, scientists are just one of many stakeholders, as you know. So we have to deal with the public. We have to explain rationally how this fits into the GMO spectrum or not. We have to explain to various people with a seat at the table, right? The scientists, the non-scientists, the regulators, the politicians, the industrials, the lawyers, the ethicists, the experts. Everybody has to voice their opinion so we know what to do, what not to do, or when to do it, or if to do it, whether to do it or not do it. Okay?
Clapper, the former the director of national security, called CRISPR a potential weapon of mass destruction. Is the military involved in the implications?
Oh, absolutely. The Department of Defense is very aware of CRISPR tech. Department of Defense has funded CRISPR research. Department of Defense has been aware of CRISPR tech for years.
Is there anything that makes this technology any more dangerous than any other technology?
No, it's potent, but there's many more dangerous and potent technologies out there for sure. If you think of, depending on what your target is, if your target is eradicating mankind, there's many better alternatives or more powerful alternatives than CRISPR. Okay. Now, if you want to hurt your neighbor, you can try to genetically edit the hell out of your neighbor. Okay, that's going to take you a higher threshold of expertise than to run him over with your car. All right, or trash him on Twitter, bully him in cyberspace. Right. So there's many other technologies that are more damaging and powerful than CRISPR. But when it comes to biology and changing genomes and weaponizing bioagents, it's pretty powerful. Okay? Now, at the same time, from a consumer standpoint, regulatory paths. Different regulatory pathways for medicine versus ag. Even within ag, plants, crops, animals, and foods are regulated sometimes in different ways by different agencies for different reasons. So check out the white button mushroom story from Penn State, or the waxy corn story from DuPont. Okay? And this is an opportunity for scientists to engage with the public, to discuss the benefits to risk ratios of various technologies. Because CRISPR can cure Duchenne's muscular dystrophy. CRISPR can cure HIV, have an infected human cell. Right? So risk to benefit ratios. And then last but not least, from a technical standpoint, again, I don't have time to go into the details here, but there are ways to harness CRISPR that are non-GMO, given today's regulations. So in many ways, there's ways where CRISPR is not even a GMO consideration. You don't even have to bring it into foreign DNA. It can just deliver RNPs, ribonucleoprotein complexes. No foreign DNA comes in. You can edit the genome without foreign DNA. Sounds like science fiction. It's just the science part. Just science, okay? All right. Now what I do sometimes to wrap it up is talk about the future of CRISPR. Looking at the past is fantastic. I've enjoyed the ride big time. Like very few others have been able to enjoy it and assess it and comprehend it and appreciate it. But in the next 10 years, I can tell you right now, CRISPR will be more and more democratized, more and more accessible. Okay? The ag applications are unraveling before our eyes. food and crops and animals. Again, non-GMO alternatives and implications from a regulatory and technical standpoint. Clinical trials underway. It's happening already. And the three companies aforementioned all have plans to go in man in 2018. Next year in the US, clinical trials for CRISPR-enhanced gene therapies and immunotherapies. Very powerful. The scale at which it's happening is unfathomable. They're like an order of magnitude every so many months. Okay? And last but not least, one of the hard parts for scientists to deal with, the CRISPR element, is not the cutting part. The cutting part has become almost trivial. Almost. The hard part is to predict the repair outcomes. To control precision repair, right, HDR to an HEJ ratio. So we're going to talk about that. We can talk about that. Now, as a faculty member at NC State, I can tell you, illustrate for all of you here, this is where some of your best spent taxpayers' money is going, in my opinion, right? So we are actively engaged in CRISPR PR with the whole world. We have a CRISPR startup company at NC State. We've generated already CRISPR alumni that are enabling the industry, people who have their PhDs on CRISPR research. So last year, right? We are licensing IP, we're creating products, and we're helping the democratization of CRISPR product out of North Carolina, for North Carolina, sometimes in North Carolina. Very, very important part of our mission. And I went online, and I'm going to give you some examples. Right? So we have great research at NC State, licensing through Intellectual Property Office, the Office of Tech Transfer. I have students that are doing work at Acknowledgements just down the street from here, about a mile and a half down the street, right there, making it happen. I gave 68 talks on CRISPR last year. It's a lot. I think this year I'm ahead of pace. Okay? We went public with Intellia. I got to ring the bell at the NASDAQ last year. That's crazy. Come on. I rang the bell. What the hell is going on here? Right? We generate CRISPR graduates. I coach alumni, and we build companies here. We publish a lot. And then last but not least, it's putting North Carolina on the map in the scientific world. Visibility. Credibility. Right? Achieve. Think and do. Those are the things that we do at NC State. So in closing, I hope that was a good overview of what CRISPR actually is from a scientific standpoint. Adaptive immunity in bacteria to fend off viruses. Right? I mean, it's kind of cool scientifically. But the reason people really care is because this can be repurposed as a machine, a molecular machine, a molecular scalpel to drive genome editing in humans and plants and animals and viruses and bacteria. You can edit any sequence you want in any organism you want, any way you want. That's it. It's that simple. It's that consequential. I hope you have a good understanding of how there are applications and opportunities across the food supply chain and medicine and agriculture and biotechnology. And then last but not least, notwithstanding how cool the scientific revolution is, media coverage, public engagement, PR, ethics, regulation, guidelines, guardrails, education, right? Those are strings that come attached with it that people like me and many other CRISPR scientists are trying to spread the gospel of on a daily basis. So I'm going to acknowledge all the people that I've been working with and getting funding from. On the left is all my collaborators for the last 10 years, notably DuPont, my former employer in the CRISPR lab at NC State. You can check us out on Twitter at CRISPR Lab. In the middle at the top is my financial conflicts of interest, because I've received money from those people or I've consulted for those people. I've been on the board of directors for the people, a scientific advisory board and consultancies for those people. and sometimes even an investor in those companies or a shareholder in those companies. In the middle at the bottom, funding and collaborations from the state of North Carolina. The NC Biotech Center has done a fantastic job at this for sure. Come to Casey Terrio's seminar and meeting in about 2 weeks down the street. USDA, FDA, CDC, and the NC Biotech Center, National Center for Biotech Information. And then on the right, last but not least, the many labs at NC State doing CRISPR research and advancing the CRISPR field for the benefit of humankind. So I'll thank you for your time, and we'll take a few minutes for questions. Thank you.